If you want to learn biochemistry, which you totally should, it can get overwhelming just looking and seeing how much information is out there. So this is an outline of what I recommend that you focus on when learning biochemistry, what I think is the most foundational and helpful. Of course, if you're taking a course, you also want to make sure that you're focusing on what the teacher is going to test you on, but in the sense of getting a good appreciation for biochemistry, this is what I recommend. First, you want to get just a general introduction to biochemistry. So be able to identify the core macromolecules and their basic characteristics. So the main macromolecules are going to be your proteins, your nucleic acids, your lipids, and your carbohydrates. And so throughout the course, you should be going one by one through each of these. But it's good to just have a general sense of these four classes from the very beginning. Because even if you talk in detail about one of them, the other ones are still going to come up along the way. You also want to identify some types of experimental strategies used to study these various macromolecules. So again, these experimental techniques are one of those things that's just going to kind of show up throughout the course. And we'll go into more detail as we get into each of those more topics and how these experiments are used. But being familiar with the basic concepts of these experiments is going to help you understand them as they come up. So their basic strategies include like structural strategies. So we'll get into things like NMR and cryo-EM and X-ray crystallography, which try to look at the 3D structures of molecules. Biophysical techniques. So these are things that typically do things like measure thermodynamics. So we're talking ITC, SPR, this sort of thing. There's also like mass spectrometry, where you can get information about the sequences of proteins and various modifications of proteins and what metabolites are like these small molecules, breakdown products or build-up products from various metabolic pathways are present in a solution, as well as various cell and molecular biology techniques. So be familiar with the concept of reporters, things like GFP or luminescence that can serve as a readout for indirect readout of something that's going on inside of a cell. Various types of probes, so like antibodies and antisense oligonucleotides that can kind of bind to various sequences that might be present in a gel, qPCR probes, various things like this. And so some of these you should encounter in the lab, and then some of them you'll see people use in the experiments that determine the knowledge, that discovered the knowledge that we're going to talk about in the course, as well as the experiments you might come across if you go and you read some of the primary literature, so the original research, which I highly recommend that you do, both historical research as well as current. And hopefully hopefully in your course, you'll get enough information that you can then be more comfortable reading those papers. And reading those papers is also going to help you greatly develop your sense of critical thinking and start to think like a scientist. What's also really important is to kind of get a sense of perspective from the very beginning. And so at least in my course, what I'm planning to do is start in the beginning with a kind of lecture on biochemical intuition and perspective. There's this really great book, Cell Biology by the Numbers. You can actually get a free version of it online, and it's an interactive version too, or you can do the PDF, very, very helpful. It helps you do things like back of the envelope estimates, get a sense of relative scale and concentrations inside of cells and in various solutions, as well as speeds. So we're often talking in biochemistry about things in isolation, but it's important to remember that things are actually happening typically inside of cells, inside of complex biological environments. And it's important to keep this perspective in mind, as well as really think about what are the relative sizes and speeds and things of what's going on, because talking in really small numbers or really, really big numbers, it can kind of be hard to get your head around that. So this book helps you do that with some more like real world examples. Speaking of things not happening exactly the same in a test tube or on a textbook page as they are in a book, as they are in your cells, it's also important to note that cells are not homogeneous. They're not like all the same, and inside of a single cell is not all the same. Cells are compartmentalized in addition to things like having particular compartments like your nuclei, your mitochondria, various lysosomes, and other membrane-bound compartments. There are also things like scaffolding proteins, which play a key role in holding together
various enzymes involved in pathways, as well as like phase separation. So intrinsically disordered proteins, as well as like nucleic acids, that can kind of cause this phase separation, or where you get this kind of like gel formed within the side of a cell. So you get this little like blob where the local concentrations of various reactants are going to be very high. And this is going to allow reactions to occur, even if there's not a high concentration of those molecules present, if you just consider the rest of like the whole cell, the cell as a whole. And so these conditions, these local conditions might differ greatly from those in bulk solution. This also comes into play when we're talking about like pH and pKa, because the local pH might be much different than that in the general cellular environment. And in some compartments, like the lysosomes, and they can actually be much, much different from those of the cell. And then you can also get things that are different in just a region of an enzyme. And so we'll talk more about that later. Another intuition-y type thing, perspective type thing, is to appreciate that much of biochemistry is probabilistic. So we can predict whether things will be unlikely or likely, but ultimately it's a number game. And so even very unlikely and or rare things can happen to a small degree. And we want to keep this in mind, especially when you're thinking about things like pKa and pH. So you might be thinking, well, that lysine is almost always going to be protonated. So how is it ever going to be able to participate as a nucleophile? But even that little bit that is deprotonated is enough to, in many cases, to be modified. And if it's in the context of an enzyme, you have other parts of the protein helping to change the local pH. So keep in mind that we're talking probabilities. And so even those rare things can happen, but things that are more likely to happen are, well, more likely to happen. Chemical foundations. So basically, biochemistry is based off of chemistry, and you need to have a solid understanding of chemistry to get a true understanding of biochemistry. Typically, when you're taking biochemistry, at least here, you'll have taken organic chemistry already, as well as general chemistry. And this is going to help lay the foundations for your biochemistry course. In your biochemistry course, however, you're not going to use everything that you learned in Gen Chem or everything that you learned in OChem, but there are particular things that you will want to refresh your knowledge of. Some of these are going to be things like redox. We'll talk more about that a lot. And nucleophiles and electrophiles, those are really important to focus on. You're not going to deal with most of the elements of the periodic table, thankfully, in biochemistry. You don't have to worry about a lot of them, but the key elements you'll want to focus on are hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. You'll also see metals come up sometimes. The key metals you'll see are like zinc and magnesium, as well as like calcium, sodium chloride, various things like that, that'll come up. But these are the main ones that you're going to see when we're talking about things like metabolism. You also want to be able to identify key functional groups. So carboxyl groups, amine groups, amide bonds, ethers, esters, acetyl groups, phosphoryl groups. These are going to help you talk about pathways as well as name molecules and see what's happening. Also going to keep an eye out for common nucleophiles and electrophiles. So you want to be looking for things like phosphate groups, things like carbonyl carbons, various things like that, and be able to be familiar with the idea of tracking electrons and doing like arrow pushing and things like that. You won't need to do it as much as in OChem, but it's still, I find it's really, really helpful to be able to track the flow of electrons to see what's going on in a reaction. So as you'll see, I don't really believe in rote memorization. And I think it's much more valuable to be able to have a greater understanding of what's going on in a pathway. And then if you need to recreate the pathway, you can kind of think your way through it. Also, refresh your knowledge of diffusion, osmosis, and tonicity, and colligative properties. So you should have learned about these things in general chemistry, and they're going to come up here as well. Thermodynamics play a really, really key role in biochemistry. So later in the course, I'm going to go into more detailed coverage, but in the beginning, you kind of want to just like focus on intuition. So get a general sense of what these terms mean, because they're going to come up when we're talking about the relative characteristics of various molecules. We'll see things like values given in Gibbs free energy, this like G and things like this. And so you want to know what that represents. So thermodynamics versus kinetics. Keep in your head this idea that thermodynamics and kinetics are different things. So thermodynamics deals with things like at equilibrium and whether things are favorable or unfavorable from an energetic scale. Whereas kinetics, this focuses on like how fast things happen. So something can be like really, really favorable, but have really slow kinetics, so not happen very quickly. Whereas something can be very quick, but it might not be very favorable. And so thermodynamics and kinetics are different things, and we'll see that we can use different techniques to measure these. A really important concept in biochemistry is the idea of dynamic equilibrium. So basically, you don't have a static equilibrium where things on both sides are just exactly the same, like all the time. And the molecules are the same on both sides of the equation. Like those molecules aren't going back and forth between the different versions. Instead, what you have is a dynamic equilibrium where molecules are constantly going forward and backwards in say a reaction, but the overall concentrations of them remain the same. So you remain at equilibrium, because that equilibrium is just looking at the bulk and saying, okay, well, how many of the substrates do we have? How many of the products do we have? Whereas it's not looking at, okay, well, is that the exact same substrate molecule that we saw when we took a picture last time? So this is the idea of dynamic equilibrium. Another important concept is that in our cells and our bodies and things like this, we're often kind of avoiding equilibrium. And so you'll see things siphoned off from various pathways. You'll see various substrates and other molecules kind of like compartmentalized and closely controlled. So by controlling the concentrations of the products, of the reagents, you're able to therefore kind of coax the reaction to go one direction or another direction. And this comes up a lot in metabolism. We're thinking about whether a pathway is going to be used in like the forward direction or the reverse direction. So when we talk about energy, we're usually talking about Gibbs free energy. And we're really usually talking about delta G, which is the change in free energy between the reactants, the products and the reactants. So basically, you want to know the difference between a negative and a positive delta G, an intuition as to what is favorable versus non-favorable, and an intuitive sense of scale of the delta G's for core biochemical reactions. So a negative delta G is going to be favorable and a positive delta G is going to be unfavorable, but this does not mean that it's going to be a likely or unlikely in terms of the delta G, because things can be really, really favorable, but they can have a really, really high activation energy. So the activation energy is going to be a really important concept. And we'll see that activation energy is going to be lowered by enzymes. And so enzymes are going to play a huge role later on. So stay tuned for that. Another thing that can make unlikely reactions happen is reaction coupling. These make the non-favorable possible. So we can do things like have reactions that break down ATP, which has that reaction has a very negative delta G, which can then be used coupled to something with a positive delta G to make it happen. In terms of the actual scale of those delta Gs, you'll want to kind of throughout get a sense of the general scale of what's the delta G you get from burning ATP and things like this. So you can know, okay, well, is that value really realistic, or is that really favorable? Is that kind of just like within the noise, or is that going to give me a lot of energy? So that's going to be helpful for that, as well as for determining whether something is likely irreversible. So it's very, very unlikely to go in the other direction, or reversible. So it can easily go back and forth. Sometimes these like irreversible reactions, or quote unquote irreversible, because nothing is truly irreversible. But if it has a really, really, really low delta G, it's like very, very unlikely to go the other direction. We call it irreversible. And so often this is happening with when we're breaking a high energy bond. So you want to get familiar with the idea of the role of a phosphate group. So basically these are negatively charged bulky groups and they're held tightly together. And so because you have all these negative charges held tightly together, it's kind of like clamping a spring a tight spring. And so just like that's, you have to kind of like apply a lot of energy and force to hold that spring clamped. You have to, it takes energy to hold those phosphate groups together, and that energy is stored in the form of what we call these like high energy bonds. And just like if you were to let go of the spring, it would like spring out. And maybe if you had a ball on the end of that spring, it would kind of like fly across the room. Similarly, if we break a phosphate bond, well, here what we're doing is we're releasing energy that can then be used to drive other reactions. And so this is one of the things that can make a reaction irreversible. And it's also something that can be used to couple reactions. Okay, so we have reactions, which we typically think of as actual like changes in covalent bonds, where we're actually changing like those true bonds. So yeah, be familiar with like covalent versus non-covalent bonds, which you should have learned about in your Gen Chem as well as in your OChem. So covalent bonds, you're sharing electrons. Those are like those true bonds. And then we have non-covalent bonds, which are more like attractions that are based on like partial charges and things like this. In biochemistry, we deal a lot with these intermolecular interactions. So not just the making and breaking of actual molecules, not just the changes in the covalent bonds, the actual reactions, the actual bonds, but these interactions. These are going to play a crucial role in biochemistry, and much of what we're doing in biochemistry is measuring those reactions, like when it comes to laboratory work and things like this. These types of intermolecular interactions are going to be things like your van der Waals, your London dispersion forces, or hydrophobic interactions. These things like induced dipoles, spontaneous dipoles, so things that aren't really involved. These aren't very strong individually, but together they can be very strong. Also like dipole-based interactions, like permanent dipole-based interactions, things like hydrogen bonds, which are a special case of those, as well as ionic bonds, which are basically just full charge interactions, attractions. And so these are kind of come up again and again and again. And at their heart, they're all kind of the same, where you have this partial charge attraction, either something that's kind of just spontaneous and not very strong, something that's more permanent, something that is with a hydrogen bond, you also have specific angles that make them stronger. And then with ionic bonds, you have those full charges. So these are going to be stronger still. And you'll see these come up again and again. And you'll want to be able to recognize what functional groups can participate in what type of bonds, and where you might see these. Something that makes biochemistry complicated compared to things you learned in OChem. Well, I mean, not that biochemistry is more complicated than OChem. Well, some parts are, but not all. But basically, some of the stuff that you learned in OChem, when you do a lot of those reactions, they're happening in like organic solutions. They're happening in things that are non-protic solutions, things where you try to avoid anything like water because water complicates things. And so, but in biochemistry, we're pretty much always dealing with watery solutions, with aqueous solutions. So we want to be familiar with water and its key characteristics, things like how it expands when it freezes, which can make things complicated when we go to freeze a protein. Things like how it forms these extensive hydrogen bonding networks, so it's really sticky to itself. Because of this, it's also, you get this thing called the hydrophobic effect, or the water exclusion effect, where if you want to stick something into water, because water has all those bonds to one another water molecules, in order to allow something into its network, it's got to like that thing. And so only things that are hydrophilic, things that have a charge or a partial charge, only those things water will want to include in its network. And if the thing doesn't have something to offer water, water would exclude it. So the water will kind of like clinch around it, minimizing its contact with that thing. Now those hydrophobic things will then be forced together, and you get these kind of like dispersion forces between those, as well as the water cinching, like the water, yeah, around them is going to cinch up and you get things like surface tension. Okay, so water can play various roles in biochemistry. In addition to kind of just being there, it can actually serve direct roles in reaction mechanisms, and it can help transfer things like protons as in various reactions. Sometimes you see direct involvement of water, and sometimes it's more just indirect. You'll see something called like specific acid-base reactions, which are actually going to be one of those things that directly involve water. Water also plays a key role in binding energies. So often when things bind, one of the things that makes it more favorable is that you get entropy of the water molecules that get displaced when that thing binds. So if you have a molecule bind to a protein and displace a bunch of water molecules, you're increasing the entropy because you're kind of releasing those water molecules to be more free. And so this can help drive the binding. Another thing about water is pH. So basically, pH, appreciate that it's a log 10 scale. So a difference of one pH unit might sound small, but that's really a 10-fold difference. And if we're talking about a 2 pH units, well, now that's 100-fold difference. So the further you can quit, as you go small amounts away from the pH, you're actually changing the original pH, you're changing things a lot. And this comes into play when we're talking about acids and bases. So when you're thinking about acids and bases, review what you learned in Gen Chem, but the emphasis in biochemistry is going to be on those used in the body and in the lab. And you want to be familiar with the autoionization of water, as well as the idea of pKa, which tells you about the strengths of acids and bases. So some rough pKa's to remember are carboxylic acids about 4 to 5, protonated amine about 9 to 10. So we'll see these in our amino acids. A thiol group, so like your sulfur, like your cysteine group is a pKa of about 10. An alcohol, a pKa about 16 to 18. Phosphorus group, about 2 for the first deprotonation and about 7 for the second deprotonation. So the pKa is going to tell you the pH at which half of the acid is in its acid form and half is in its conjugate base form. And so you want to have a good sense of being able to say, okay, well, I'm a pH above the pKa. What this means is there's going to be fewer protons around, and so my molecule is less likely to be protonated. So less than half of it is going to be protonated. But if I'm below the pKa, well, now there's tons and tons of protons available, and so more than half is going to be protonated. And for each 1 pH unit I go from the pKa, now it's going to be a 10-fold increase in the one that is the major form. And so we can actually put this into an equation, and we can use this Henderson-Hasselbalch equation to calculate how much is protonated versus non-protonated at a certain pH. And this is going to come in really, really handy when you're learning about those amino acids. So you can use the Henderson-Hasselbalch equation to calculate the amount of an acid and its conjugate base, and to calculate the amount that you need to mix in order to get a certain pH. And this will come in handy when you prepare buffers in the lab. And know like key buffers used in the body in the lab. So focus on the bicarbonate buffer system and the phosphate buffer system. Okay, now let's get into structural biochemistry. So when we talk about structural biology, we're typically talking about like the 3D structures of big macromolecules, typically proteins, sometimes like nucleic acid protein complexes or big nucleic acids. But here I'm using structural in the broader sense of the word and talking about really the chemical structures of these molecules as well, even the small building blocks. But first, I just want to give you an idea of some of the intro to structural biology techniques. So have a concept of the idea of maps and models. So when you're looking at those pretty pictures with all those like cartoon figures and those ribbons and those sticks, those are just models. Those are structural models of the 3D makeup of the molecules. But they're based on experimental data that gives you these kind of like maps, which basically show you kind of like a fuzzy outline of the molecule. And then scientists have to build in the model into that. And so there are limitations to these, and it's not like exact. And so be familiar with this concept of like resolution, where basically some regions can be more fuzzy and some regions can be so fuzzy that you can't actually see the molecules in that part, or you can't actually see the atoms in that part of the molecule. And so you might see actual missing regions. So don't worry about the details, just know that there's this concept of resolution. The higher the resolution, so that's going to correspond to lower numbers, because the resolution refers to how close together two things can be, and you can still see that they're different things. And so a higher resolution is going to have a lower number. That's going to mean you can kind of, it's less fuzzy. You're more certain about the structure in that part. Whereas something with a lower resolution is going to be more fuzzy. And so you're going to have a higher number, and you're less certain about that area. So that's just something to keep in mind. And you might not have this emphasized in your course, but coming from a structural biology lab, this I think is a really important concept that a lot of people don't really know about. So how those fuzzy maps are generated, these are basically techniques like X-ray crystallography, cryo-electron microscopy, nuclear magnetic resonance, or NMR. And then scientists determine these structures and they put them in this database called the PDB. So get familiar with using that PDB. If you want to go further into looking at those structures, familiarize yourself with Chimera X and or PyMol. If you want to play around with proteins in the lab, or you want to understand how scientists play around with proteins in the lab, know the basics of protein purification using column chromatography. So be able to identify which of the main types. So affinity chromatography, when we're talking about if we tag our protein with something like a his tag or a strep tag, GST, MVP, something like this, what we can use as an affinity tag to purify it out specifically based on that tag. Talk about ion exchange, so anion or cation exchange, which separates based on charge, or size exclusion, which, as the name suggests, separates based on size. And this is also called gel filtration. And so be able to identify which of these types you could use to separate proteins with given properties. So if I were to say, give you a couple of proteins and tell you about the various properties, be able to tell me what technique you could use to separate them. Now let's talk a little bit about the underlying protein structure. So there are basically 4 levels of protein structure. And so start with an overview of them, and then refer to these as you build up from amino acids to protein complexes. So the first level, the primary sequence, is the primary structure. This is just your sequence of amino acids. The secondary structure, this is going to be structural motifs involving backbone interactions. So you get hydrogen bonding interactions between the backbones of the amino acids. You get things like beta strands and alpha helices and turns, etc. Tertiary structure, this is going to be the overall structure of a single chain, including structural features formed by interactions involving side chains. And then if you have a quaternary structure, which not all proteins do, this is a protein that involves multiple chains. So each of those chains is basically translated like it was, you can basically have different subunits of a protein that come together to make your overall protein. And so they'll have multiple chains that are translated individually, but then they come together to give you this quaternary structure of your protein. So that's another thing that isn't always quite intuitive, is that proteins can be made up of multiple chains. Okay, but at their heart, proteins are made up of chains of amino acids. So start by thinking about the amino acids. Be able to draw the core amino acid backbone, so that generic backbone, complete with stereochemistry. So which direction are those various things sticking out into space? When it comes to stereochemistry with amino acids, we talk about L versus D, and how the L amino acid is what our bodies use. And I recommend playing around with molecular modeling kits, so these like MollyMod kits. And just a quick note on L versus D. So when you learn about L and D in your organic chemistry class, initially it's like L is for lever rotatory, so turning light to the left, and D is for dextrorotatory, so it turns light to the right. And that's the case sometimes, but not always in the with biochemistry, with amino acids, because the L and the D are actually based on how the molecules are structurally related to glyceraldehyde. And so it's not actually going to represent which direction it rotates light, because some of these might be actually lever rotatory and some are dextro rotatory. So that's not really what the L versus D tells you. It's just based on the structure related to glyceraldehyde. So that's just a little fun fact. But anyway, appreciate that amino acids are almost always in their zwitterionic form. So something that's a zwitterion, it has like a positive part and a negative part, but they cancel each other out. And so in the case of amino acids, you're going to have your carboxyl group almost always deprotonated and your amino group almost always protonated. And so you're going to have a zwitterion. And so draw them this way, the non-ionized form where you would have the actual carboxylic acid, so it actually has the hydrogen attached still, it's protonated, and the amino group is deprotonated. That is not physiologically relevant or really plausible, because if you think about it, if you have a pH that's low enough that the carboxyl group is going to be protonated, well, you know that amine group is going to be protonated as well. So don't draw that non-ionized form. I'm not a fan of rote memorization, but one area where I do think memorization is important is memorizing the amino acids. So ideally, you'll memorize the structures. So I'm planning on making my biochemistry, for biochemistry students memorize drawing the structures. The biology structure students, I'll probably just be able to recognize the structures, but I wasn't made to be able to draw the structures. And so I always regret that I wasn't, every time I go and try to draw out a tryptophan and have trouble. But the structures are going to be really important, as well as the 1 and 3 letter abbreviations, so that we can talk about amino acids without having to go look them up. And if we see a letter on a map, or if we see the mutation notation where you see something like A29Q, you'll be able to kind of think through what consequence that mutation could have without having to go look up what these various things are. So know those, and then memorizing those will let you focus on the more important and fun stuff, the actual properties of those amino acids. So there are a lot of ways to be able to classify amino acids, and you want to be able to classify them by multiple parameters, including their polarity, their hydrophobicity, their charge. For the ones that are ionizable, so know the relative pKa's of the ionizable side groups, and be able to calculate what proportion of the free amino acid form is on average protonated at a given pH. So this is what we talked about before, but also note that the local context within a protein can greatly influence the actual pKa and the actual pH. So this is why, one of the reasons why we can get the deprotonation of lysine in the context of like an active site of an enzyme, and therefore have a strong nucleophile that can drive a reaction. Also be able to classify amino acids based on their size. So whether they have branches in their chain, are they aliphatic, so like chains, or aromatic, so rings? Are they weirdos? What makes glycine and proline unique? Also characterize them by the presence or absence of functional groups, and thus the types of reactions and interactions they might participate in. Do you see things like hydroxyl groups? Do you see things like amide groups? Later in the course, you should also be able to classify by essential versus non-essential when we're talking in the dietary sense. So essential meaning that you have to get them through your diet pre-made, or non-essential meaning you can make them from other metabolic processes, so your body can make these kind of like from scratch. Also be able to classify based on like ketogenic, glucogenic, or both. Okay, so we've gone from amino acids, and now let's start piecing them together, and we can piece them together using peptide bonds. You want to get a strong appreciation for the peptide bonds, especially its partial double bond character, which is going to limit the rotation of the backbone. Be able to identify the phi and the psi angles, which are basically the points on the backbone where you actually can do some rotation, but know what limits that rotation. So like steric hindrance between your different R groups, so the side chains that stick off. Be familiar with Ramachandran plots, which kind of plot out the angles of these phi and psi. And so we'll see when we talk about the secondary structure that you can actually see various angles will be favored in helices and sheets. And you can see those kind of like cluster on that Ramachandran plot. Identify cis versus trans peptide bonds and explain why trans is almost always favored. This is where our steric hindrance is going to come into play again, because you don't want those side chains like whacking into each other. Also have an overview of how peptide bonds are actually formed in cells via translation. So in your textbook, you might see this like simple condensation reaction where these two things come together and you lose water molecules. But actually how it happens in your cells is a stepwise process. You don't need to know the details, but just take a quick look at the process and return to it later to get an appreciation of it. And so, yes, you do lose the equivalent of a water molecule, but they're not actually just like coming together like that. You first have it transfer from like a tRNA. Okay, so you put a couple of amino acids together and you keep putting more and more together. And eventually they're going to kind of start forming a cooler structure. So with the secondary structure, these are going to be motifs like beta strands, which are kind of like these crinkly strands, and then beta pleated, these zigzags, and then beta pleated sheets where you kind of have these strands come together and you get this kind of like accordion fold looking thing. And they can be parallel, so the strands are going in the same direction, or anti-parallel, those strands are going in opposite directions. You can also get alpha helices, so those kind of like coily looking things and turns. These are going to be based on the hydrogen bonding interactions of the backbone. And because the backbone is generic, even with, well, you could have different combinations of primary sequence give you these same characteristic patterns where you have because you have that repeating nature of the hydrogen bonding forming groups. Those amine groups and those carboxyl groups are going to be evenly spaced throughout, no matter what the side group is. So you can get these characteristic structures. See how their characteristic phi and psi angles show up on Ramachandran plots, as we were talking about before, and identify those backbone interactions that are holding those structures together. So see which of, like, okay, well, I have this amino acid, well, which one is it going to actually be bonding to? And so like count, okay, well, it's how many farther away, and you'll see that although they're not direct, the ones that are close to each other in the 3D space might not actually be close together in the sequence. Be able to appreciate the faces of secondary motifs. So basically, if you have one of those helices, one side of it might be exposed to the surface, might be surface exposed, might be exposed to water, whereas the other side might be kind of more buried in the protein. And so the amino acids that want to be on either side of that helix might be very different. Explain why certain amino acids prefer to be in certain motifs, and predict which type of a motif that given amino acid might prefer. And so, for example, long and flexible tends to prefer an alpha helix, where they can kind of stick out and have all sorts of room, where they can kind of like wiggle away from one another. Basically, these long skinny, they're not going to be clumped together, because remember, in this space, they're kind of going to, the ones that are next to each other are kind of going to still be next to each other in space. And so if they're big and bulky, they kind of hit each other. But if you have something like a beta strand, well, now these things can be on like opposite sides, and you can get things where the short and bulky are going to be able to be next to each other. And so these tend to prefer the beta strand. Glycine and proline, they don't like either. Glycine's too flexible, so it doesn't like being confined like that. And proline's too rigid, it's held in a specific shape, which with the angle doesn't conform to the beta strand or to the alpha helix. And so you also see that when you look at a Ramachandran plot, glycine and proline are going to not cluster typically with those other guys. Also appreciate the presence and the roles of intrinsically disordered regions, or IDRs. So these are basically regions that don't have a characteristic secondary structure. They're more loosey-goosey. But that doesn't mean they're not important. They can form scaffolding roles to kind of hold things together. They can act with phase separation, so making those jelly globules and stuff like that. Also be familiar with the technique of circular dichroism spectroscopy, or CD. Don't worry about the details, just know that it's a technique that can help show the relative proportions of alpha helices, beta sheets, and IDRs in a protein. Okay, tertiary structure. So now we're talking about kind of like the overall 3D structure of a protein, which involves the interactions with the actual side chains and not just the backbone. So you can have side chain interactions. You can have side chain backbone interactions and things like this. All of this is going to help you form your tertiary structure. So this involves a single chain, but it's overall 3D shape. So you want to identify those types of interactions holding the structure together and which amino acid residues participate in which. Be familiar with the concept of domains. So we can talk about structural domains, which are kind of regions of a protein that can fold independently. We can also talk about like functional domains. So what parts of the protein do what. Appreciate how the amino acids that are far apart in their primary sequence can actually be close together in the structure. And predict which amino acids would want to be where in a protein. So if you have hydrophobic amino acids, those would want to be inside the protein. If we're assuming we're talking about a cytosolic protein, so one that, or an extracellular protein, basically something that's in a watery environment. And something that's hydrophilic will want to be surface or solvent exposed. Be also familiar with disulfide bonds, so we can get these forming between cysteines. And you're more likely, know where they're more likely to find them. So these are more likely to be found in extracellular proteins because glutathione maintains a reducing environment inside of cells. And also because these disulfide bonds are going to help certify those excreted proteins. Okay, understand how heat, chemical denaturants, and reducing agents can unfold the proteins. And also appreciate that proteins can have multiple conformations or shapes that they can switch between. So we can call these like conformational changes when they go between those different forms. Sometimes this can happen if they get post-translationally modified with like a phosphorylation, or if they bind to some other molecule. Sometimes but not always, you'll have a quaternary structure, which is basically where you have multiple chains coming together. So be able to appreciate how a single protein may contain multiple chains and know the terminology for these multimers, things like dimers, trimers, homo, whether they're like all the same chain, or hetero, whether you have different chains. Also identify types of interactions holding these structure together. So sometimes this includes the disulfide bonds, but not always. So also you want to be able to think, okay, well, if I were to use a reducing agent, which would take apart those disulfide bonds, would they take apart the dimers? And so this will come into play in the lab if you're thinking about what would happen on, say, your SDS page gel. If you have a dimer that's formed with a cysteine bond and you don't add your reducing agent to your buffer, you can have those run together. But if you add the reducing agent, they should come apart. Okay, so that was post-translational modifications can throw a wrench into all of that. Cause conformational change and things like this, cause changes in protein's activity. So what am I talking about? Well, translation is the process of making a protein, and so you want to have a general overview of that process. And if you have time, go into it in much more detail because it's really, really cool. But just know the basics and for general biochemistry purposes, and then know that that's piecing together the amino acids like in their original form, but then after that protein is translated, those amino acids can get modified. Things like phosphorylation and glycosylation and lipidation that we will return to later in the course when you're talking about carbohydrates and lipids and things like this. Not all the amino acids can have each of those modifications, so identify which of the amino acids can get which type of modification. And know some of the roles that these modifications can play. And know that these modifications are typically added by specific enzymes and specific locations, and that those enzymes can be regulated to help regulate the proteins. Okay, protein folding. So we'll talk about protein folding both in vitro and in vivo. So in vitro, so kind of like outside of a natural context in a test tube or something like this, the primary sequence is going to be of primary importance. There's this thing called Anfinsen's hypothesis, which is basically that you can know the entire folding of a protein based on just the sequence itself, and that the sequence totally dictates the folding. But how proteins actually fold in cells is more complicated because the conditions are going to be more complicated, and you have things like chaperone proteins that are going to help the proteins fold properly. There's this idea of a folding free energy landscape you should be familiar with, where basically there are various energetically favorable shapes that a protein can take, and the most favorable one would be what the protein would ultimately take. However, the protein can get stuck in kind of local minima. And one of the roles of chaperones is to kind of prevent the protein from getting stuck in those local energy minima, where it would have to have energy to go like over the hill and find that better valley. So be familiar with that concept. Also be familiar with the idea that protein misfolding can play a large role in various diseases. Things like Alzheimer's disease, ALS, various disorders. Also be familiar with the idea of prions, both infectious and non-infectious, which are basically proteins that can, like they misfold and then they can cause other proteins to misfold and aggregate along with them. Be familiar with some of the biochemical techniques for analyzing folding, so chemical and thermal denaturation assays. So in my course, we're going to do like a chemical denaturation assay and measure the denaturation using like intrinsic fluorescence. So that'll be fun. Also be familiar with the basics of protein structure prediction, so like AlphaFold, including its strengths and its limitations. So basically it does have limitations. It doesn't take into account things like those post-translational modifications. There are various versions that can do things like try to predict interactions and things like this, but these still are limited. And so just kind of have an idea of the current state of knowledge. PI, isoelectric point. So understand what this is. So this is like your pKa, but it's at the protein level. So it's going to be the pH at which half of the protein is electrically neutral overall. And so you can have some of those groups be positive and some of those groups be negative, but overall you're going to get a neutral protein. Be able to calculate the pI of a short peptide if you're given the pKa's of the component amino acids. But when it gets to a really big protein, you're typically just going to use some software to do this, like exposé prop param. Be familiar with how this PI comes into play in the lab. So how can we take advantage of the different charges of proteins with methods like isoelectric focusing or IEF and ion exchange chromatography, as well as how we kind of in our page gels, we typically run an SDS page. And one of the purposes of that SDS, it's not just to denature the proteins, it's also to give them all a uniform negative charge because proteins have different charges. And so if we use charge to separate these proteins, then we're going to, which would be like isoelectric focusing, they're going to different proteins with different charges would travel differently and not travel just based on their length. So that was the protein structure. And now we'll talk about protein function. So one of the fundamental goals is to be able to see and explain how a protein structure, so how its form relates to its function. So some proteins serve like fully structural roles. So be familiar with that concept of scaffolds that we talked about before, and also appreciate how proteins shape things like your cells and provide like strength to your muscles and all this stuff. And some proteins like bind other molecules. So we call the things that a molecule binds a ligand. So proteins can bind to ligands, and we have various ways that we can characterize the binding. So you'll want to know binding kinetics and thermodynamics. So remember, kinetics, we're talking speeds. Thermodynamics, we're talking about like at like equilibrium and how energetically favorable or unfavorable is a reaction, regardless of how fast it actually occurs. So understand this term called KD, which is the concentration at which half of the thing is bound and half is unbound. Be able to derive it and see where it comes from. Be able to identify it on graphs. So if I give you a graph showing binding data, can you show me the KD? Be able to compare KDs and recognize that a higher KD corresponds to a weaker interaction because you have to have more of a thing for it to actually have half of it bound. So each time it runs into it, it has a chance of binding. And the more substrate you have, the more ligand you have, the higher the chance of them coming together. And then the more they like each other, the higher the chance of them actually staying together. So the more of the partner that you have to add in order to have enough interactions for them to stick together, the weaker that interaction is going to be. So a higher KD is going to be a lower affinity, and a lower KD is going to be a higher affinity. And so deriving this can help you see where it comes from. So do that at least once, I suggest. Also be familiar with some of the biophysical techniques that we can actually use to measure binding. Things like ITC and SPR. We're going to do ITC in one of my course labs. It should be fun. Describe how to set up an experiment to measure binding and calculate KD from binding data. So again, this is something that should be reinforced in your lab, hopefully. But know how, okay, well, why would I want the concentration of this thing to be really, really low? Things like that. And what range of value should I use? So those are going to be important things as well. Be familiar with the potential for cooperativity. So basically, you can have things where if a molecule binds to multiple other molecules, the binding of a first molecule might influence the binding of the second molecule and make it easier. So this is represented with these things called like hill plots and equations. So recognize cooperative binding on a graph. You'll see things like an S-shaped curve, depending on the type of graph you're looking at. Hemoglobin is a great example of this. It has multiple subunits that each can bind to oxygen, and the binding of oxygen to one of them kind of makes the binding of oxygen to the others easier. So this is a great idea of cooperativity. And hemoglobin is often one that's focused on in biochemistry classes. It's also a great example of how pH affects the binding. And this is something called the Bohr effect, as well as how BPG affects hemoglobin binding oxygen, et cetera. So know the hemoglobin example because that's a really, really great example of various forms of regulation and allostery. So we're binding at one place affects another place, all sorts of cool things with hemoglobin. Also understand the concepts of avidity and affinity. So affinity would be like a single site, the strength of that binding. And avidity is if you have like multiple sites kind of helping anchor things together. So if you think about a zipper, if you only had one of those teeth, it might not be very strong. But if you have a bunch of those teeth together, if some of those teeth were to come out to break or come off, it would still have a high, it would still be really sticky. And those pieces that had kind of like come off for a second can now come back on because you have a higher local concentration. So that's your idea of like avidity. One of the places that shows up a lot is in like antibody binding. Okay, enzymes. So now we're talking about real activity stuff. Appreciate that enzymes catalyze reactions in both directions. So they're not just a single direction. If something is more favorable in one direction, the enzyme, yes, it will catalyze it in that direction, but it still is able to catalyze it in the opposite direction as well, because what's happening is really you're just lowering the activation barrier. You aren't tipping the balance. And so the enzymes can lower the activation barrier, make it easier to get over that hump and get the reaction happening. And when they do so, they don't get used up in the process. And so that defines an enzyme. Explain the induced fit hypothesis and the idea of a transition state. So the idea that you might have heard of like this lock and key theory, where basically the substrate, so the starting material for an enzyme, like perfectly matches the active site of the enzyme like a lock and a key. But really it's more like an induced fit where it kind of matches. It matches pretty well, but not exactly. And so the thing binds and it kind of gets tweaked into this unnatural state, which is more less favorable state. This state that's like prone to like then reacting and breaking or whatever, this transition state. And so the enzyme is actually going to be stabilizing this kind of awkward transition state to promote the reaction actually happening. It's stabilizing that unstable part so that the reaction is more favorable and less likely to just like go backwards. So by doing so, it's kind of lowering the activation barrier. So basically, you can identify some of the ways that they do this. When I said go backwards, I didn't mean like, yes, it can catalyze the reaction in the reverse direction by doing this as well, but you're still making it easier for the forward direction to go forward. Okay, so some of the ways that enzymes are able to do this, they can hold molecules together, they can alter the local pH, they can give and take protons or electrons, provide cofactors, etc. So be familiar with the EC classification system for enzymes and be able to explain what enzymes of each class do. Be able to give examples of enzymes in each class. And I highly recommend going through and in detail examples of the various enzymes, not trying to memorize them, but really just trying to understand what's going on. Understand and give examples of acid-base catalysis, both general and specific. So specifics where we're actually involving water. General, it can be like a different acid or base. Be familiar with types of cofactors. So in the metabolism section, we'll encounter several core cofactors. And when you get that, you should be able to recognize what role they play and predict what cofactors might be used for what type of reactions. So like NADH, if we're talking about reductions. Also explain the roles metals can play in enzymes. There's some classic reaction mechanisms to know, such as like a cysteine protease. This is a really great reaction mechanism to know. Understand enzyme kinetics. So now we're talking speeds. When we talk about enzyme kinetics, we're often talking about Michaelis-Menten kinetics. Get comfortable doing calculations with these and make sure you include the units. Derive this equation at least once to get an appreciation for it, similarly to with our KD. Explain the mathematical definition and, more importantly, the practical meaning of these terms, Km, Kcat, Vmax, and your specificity constant, so Km divided by Kcat. Be able to interpret a Lineweaver-Burke plot. So this is also called like a double reciprocal plot. And know its strengths. So it's easy to get values for Vmax and identify types of inhibition. You don't need fancy software to fit the curve, so these can make it helpful. But it also has limitations. So you basically have uneven weighting of points with more emphasis of measurements from lower values, which you might not have be as accurate. And this is going to skew your graph. So if you have access to something like Prism, that's going to make it so you can make a more accurate value, get more accurate values. Compare and contrast Km and Kd. So Km is KD we talked about earlier when we're talking about affinity, and Km. So this is basically going to be kind of like that, but for an enzyme context. And it's not going to be exactly the same because they're kind of like two ways out where something can unbind or it can also get transformed into products. Distinguish between pre-steady state and steady state kinetics. Describe how to set up an experiment to measure binding and calculate the KD from binding data, or Km from binding. I forgot to alter this, but set up an experiment to measure kinetics and calculate Km, and this will be reinforced in the lab. Explain the terms activity and specific activity. So when we're talking about specific activity, we're talking about like the activity in terms of how much like mass-wise you have. So appreciate how specific activity should increase as you purify an enzyme-containing solution. Also appreciate how this thing called like an activity unit or U can really be anything a person defines it as long as it's defined. And recognize how this comes into play in the lab when you get those restriction enzymes or something and it's talking about units and you're like, what the heck is a unit? And then you have to go look it up in their brochure. Know the types of enzyme inhibition, so competitive, uncompetitive, non-competitive, mixed. This should be reinforced in the lab. Be able to identify what effects they have on kinetic parameters. Be able to recognize evidence on them from plots and be prepared to draw the effects they have on plots. So if I were to show you a plot, will you be able to and say, okay, well, is there an inhibitor present? So we have this inhibitor present. What type of inhibition is occurring? Or if we have this type of inhibition, what would we expect to see on the graph? Be able to do those sorts of things and be able to give examples of different types of inhibitors. So that was proteins. Now let's move on to carbohydrates. So you want to know the defining features of carbohydrates and be comfortable with the symbol notation that they, if you have a key provided, so you don't need to memorize what color triangle or color square corresponds to what, but be familiar with the basic symbol notation that's often used to represent carbohydrates. Much of the stuff on carbohydrates should have been covered in OChem. So in my class, we're going to focus on glycoconjugates, but you should also know the following. So for monosaccharides, those individual sugar units. Be familiar with Haworth and perspective formulas. So where you have those kind of like 3D rings and Fisher diagrams. For linear sugars, be able to describe structures of as aldoses or ketoses by their number of carbons. So you're talking a pentose or a hexose. By the number of sides of the ring structure, you're talking about pyranoses or furanoses, things like this. Appreciate that there is a core set of monosaccharides, but these can also have derivatives such as amino sugars, deoxy sugars, and acidic sugars. Disaccharides, when you have two monosaccharides joined together. Know that these are joined together through an O-glycosidic bond to be able to identify that bond. Know components of two of the main disaccharides, so know that lactose is made up of glucose and galactose, and that sucrose is made up of glucose and fructose. And also be able to identify what types of sugars these are using these descriptive terms. Polysaccharides, when we have multiple of those monosaccharides come together. Appreciate storage versus structural functions and what characteristics are ideal for what purposes. If you have something that's really nice and compact, that might be good for storage, but if you have something that's long and sturdy, that'll be better for your structural forms. Compare and contrast glycogen, starch, cellulose, and chitin. And yes, this next stuff will probably be new. You probably didn't cover this too much in OChem, but glycoconjugates. Know the type of glycoconjugates, so glycoproteins, proteoglycans, glycolipids, etc. Be familiar with how glycosylation and glycation occur. So glycosylation is where you have those glycans added, those sugars added by an enzyme. And glycation is where basically it's non-enzymatic. You have a bunch of these like reactive sugars in the around. And so this can play a role in like diabetes and hemoglobin modification. Identify some roles of glycoconjugates in biology. Be familiar with N and O-link glycosylation. So basically you can have that sugar chain attached to a nitrogen or an oxygen. And so identify where such glycation can occur. So which amino acids can be N or O glycosylated. And where does that happen in cells? Nucleic acids. Be able to distinguish between nucleosides and nucleotides. So with nucleotides, well, here you have phosphate groups attached. Nucleosides, you just have the sugar and the nucleobase. Speaking of those nucleobases, be able to recognize the structures of the 5 nucleobases and be able to name nucleotides and nucleosides. So if I show you the structure of one of them, can you tell me its name? Also be able to draw the hydrogen bonds that form between the bases. Show me the mechanism for phosphodiester bond formation and how it's catalyzed by polymerases, including the roles of magnesium. This helps you understand why we need magnesium in that PCR reaction. Explain why RNA is less stable than DNA. Know how nucleic acids are affected by pH. So what happens to DNA or RNA at a low pH or a high pH? Appreciate the roles of nucleic acid methylation. Describe the levels of nucleic acid structure. Compare and contrast typical structures of DNA and RNA. And appreciate that that even that characteristic structure of DNA, that double helix, it's actually going to be wound up around proteins called histones, and those histones can be modified, which can affect the accessibility of DNA structure. Also be familiar with the gist of various DNA sequencing technologies, so how they work, what are their strengths and limitations at this point in time, so short reads, long reads, that sort of thing. Lipids and membranes. Be familiar with the general structure of lipids, phospholipids, and steroids. Appreciate that lipids can be modified and moved around. So they can kind of like flip between those different sides of the membrane, and they can serve roles in signaling. And this will be returned to later on in the metabolic signaling portion. Recognize that lipids aren't confined to the plasmid membrane. So we often think about, okay, well, we have these cells, we have this lipidy membrane around them, but there's also a bunch of lipidy membranes inside of the cells. So you have various compartments, and these compartments are often interconnected. Appreciate that biochemical membranes are actually largely proteins. So yes, there's a lot of lipids, but there's also a lot of proteins in there. Speaking of these proteins, identify the types of membrane proteins. So you can have like peripheral, which are kind of just hanging out on one side, or integral, where basically they're actually going through the membrane. Identify various roles that these membrane proteins can have with transport or signaling. Explain their structural characteristics, and know the basics of how they are produced. So by structural characteristics, like these are going to be kind of like inside out compared to our other things. So they're going to have, in the membrane spanning part at least, they'll have those hydrophobic parts exposed to the surface because they're going to be exposed to the lipids. And if you have something where you have a hydrophilic channel in the middle, well, then you're going to have, you have a characteristic structure where you have that hydrophilic part kind of facing into the channel, and providing a passway through. So know the basics of how they're produced. Because they have this kind of like weird inside outness, they have to be produced through a special pathway. And also be familiar with strategies used to study them in the lab, such as detergent extraction. Speaking of detergents, explain how soaps and detergents destabilize membranes. Identify the ways in which molecules can get into cells. We talked a little about how proteins can form pores. Sometimes proteins can form polars that things can just kind of like flow through. Sometimes you actually get active transport where proteins have to actually spend energy, enzymes like spend energy to get things into cells. Know what molecules can get freely into cells. Be familiar with the concepts of tonicity and osmotic pressure. So identify hypotonic and hypertonic situations and predict how water and or other molecules would flow in various situations. Think about why we don't give people like IV water. Instead, we would give them IV saline. This has to do with this whole tonicity thing. Appreciate how getting things into cells is a major holdup for many potential therapeutics and things like this. And be familiar with some of the strategies that we use in the lab or in therapeutics to get information. Or I guess for here, it's transforming or transfecting cells, so getting DNA, nucleic acids into cells, but also know strategies for getting like proteins into cells, things like lipid nanoparticles. This will help you connect to the bigger picture. Okay, so now we've talked about the structure of these molecules. Now let's talk about how we can kind of like bring things together. Biochemical signaling. Know classes of hormones and be able to give examples of each. So we're talking things like peptide hormones, steroid hormones, all that good stuff. Know which can and can't get into cells. So some of these hormones are going to act by binding to receptors on the surface and signaling downstream pathways, whereas things like steroid hormones can actually go all the way in and get into the nucleus and directly influence things. A lot of these signaling pathways are going to involve kinases, which are going to add phosphate groups, and phosphatases, which remove those phosphate groups. And these allow for these pathways to have like single signal transduction. So send the signal from the top, from outside the cell to inside the cell, and you get like signal amplification, as well as second messengers. So kind of molecules that can take over the passage of the signal inside of the cells. So be familiar with those concepts and be able to explain them and their roles. Be familiar with various forms of receptors. Know the basics of GPCR signaling and receptor tyrosine kinases. Explain how mutations in signaling molecules and signaling pathways, so like these kinases, can lead to diseases such as cancer. You kind of get uncontrolled signaling, especially with like growth hormones, pathways, and things like this. You can get uncontrolled growth. Be familiar with nuclear hormone receptors. So basically where those steroid hormones can actually go all the way in and influence things directly. And also appreciate the role of serum proteins in the transport and bioavailability of molecules in the bloodstream. Okay, metabolic pathways. So some of the core concepts is be able to define the terms metabolism, so the making and breaking of molecules, anabolism, which refers to the making of molecules, catabolism, which refers to the breakdown of molecules, and anaplerotic, which is basically reactions that produce intermediaries of a pathway for use. Appreciate that metabolic pathways are really more like webs or interconnected subway systems, so you have things being siphoned off in various directions. Appreciate that some pathways are limited to particular tissues, so commonly the liver, because the required enzymes are only expressed there. Appreciate that different tissues and or cellular compartments, such as the mitochondria, use different versions of enzymes that may differ in their regulation and or their activity. So this whole metabolic stuff can get really overwhelming if you try to just memorize it. Instead, I recommend that you focus on learning these things. Start by learning and reviewing the fundamental underlying principles. Then when you're faced with a pathway, examine the starting and ending materials and identify what needs to change. Then walk through the pathway and track the flow of electrons. Be on the lookout for functional groups and cofactors. Look for those nucleophiles and those electrophiles. Understand the purpose of each step in the pathway. Why are you changing something? Are you adding a phosphate group to make something kind of like add a flag, make this more energetically favorable and get a reaction to happen at a specific place, something like this. Recognize which steps will be thermodynamically easy, so like favorable, or hard, non-favorable. And note that these steps may have alternative pathways in the reverse direction. Note steps at which intermediates can join in or be siphoned off into other pathways. So this is going to be, again, going back to our idea of kind of more like a web or a subway than actually just like a pathway. When you're learning, be forewarned that different sources might use different abbreviations for the same molecule, so be on the lookout for that. Okay, so now let's get in a little more detail. So those fundamental principles, thermodynamics, we're going back to this concept we brought up before, but here in more detail. So review that and think about reversible versus irreversible reactions. To be able to explain the difference. So if something is so favorable, it's unlikely to go backwards. And this can happen if, say, you burn ATP, and especially if you break an ATP and then you break down that that pyrophosphate into orthophosphate and things like this. So if you break those two phosphate groups apart into single phosphate groups, well, now you're getting a bunch of entropy and things like this. So that's going to be very energetically favorable and very unlikely to all kind of like piece back together and go backwards. So that's an example of like an irreversible thing. So be able to explain the difference between these and predict whether a given reaction will be reversible or irreversible. Explain the roles of irreversible reactions. So these can do things like make commitment to a pathway because it's irreversible. Trapping of intermediates. So if you put a phosphate group on something that could freely enter the cell, well, now it can't go through anymore. It can't just like flow back out because now it's got that charge and the membrane isn't going to like it. Explain how concentrations of products and reactants can be regulated to drive reactions in particular directions. So review that Le Chatelier's principle, because this is going to come into play where even if the reaction isn't that favorable in the forward direction, if you don't have anything around that can go in the backward direction, but you have a ton of stuff around that can go in the forward direction, then you're going to be able to go in the forward direction. Know that, so enzymes are going to play key roles in making metabolic reactions happen. Know that enzymes often do this with the help of molecules called cofactors. And so cofactors can be things like metals, in which case we have metalloenzymes, or they can be things like cofactors, coenzymes, which we typically think of as like organic molecules, so often like vitamin derived. Know the core coenzymes and what type of reactions they'll be used for. So learn them as you go along, but these are the key ones that you should remember. And when you see these, you should think, oh, this is this kind of reaction. Or if you have one of those, like, okay, I have this kind of reaction, what type of cofactor might I need? So NADP, NAD or NADP plus, or FAD, these are often involved in redox reactions. So you see these a lot in energy generating pathways because they act as electron carriers to take electrons to the electron transport chain. PLP, pyridoxal phosphate. This is involved with amino group transfer. So this comes into play with amino acid metabolism when we're kind of breaking down amino acids or switching between amino acids. TPP or TDP, these are various abbreviations for thiamine pyrophosphate, and this is involved with decarboxylation. Biotin, this is involved with carboxylation. Tetrahydrofolate or THF, which comes from folic acid. This is involved with one carbon group transfer. And so you see this a lot in anabolic pathways. So when we're building molecules up. Coenzyme A, CoA, this is involved with acyl group transfer. So you might see like acetyl-CoA a lot. So this is seen in acetylation reactions. So you'll see a lot of redox reactions, especially when it comes to things like cellular respiration, which is where we're breaking down like glucose to get energy. So with these redox reactions, we have, you want to review those principles of reduction and oxidation, identify whether a reaction is a redox reaction. If it is, identify which molecules are being reduced and which are being oxidized. Know those cofactors that are commonly used in these reactions, and recognize how reactive oxygen species, so these ROS, can cause damage to biomolecules if not controlled. So recognize how you can get, say, oxidation of a cysteine. Okay, cellular respiration. Start by getting a good understanding of the general overview of the process. So I feel like this is much more important than memorizing the details of each of the reactions. But if your teacher wants you to know those details, of course, learn those details and memorize them. But if you don't need to memorize them, then just focus on this general overview and really appreciating what's going on. Identify places where products of various energy sources join in, and appreciate how energy is being transferred in the passing of electrons ultimately to oxygen to generate ATP. Identify the key parts of the overall cellular respiration pathway, so glycolysis, pyruvate oxidation, the citric acid cycle, which is sometimes called Krebs cycle or the tricarboxylic acid cycle, and oxidative phosphorylation. Know where each of those parts takes place. Is it taking place in the cytoplasm? Is it taking place in the mitochondria? If so, is it in the mitochondria? Where in the mitochondria? Is it in the cortex? Is it in the matrix? This sort of thing. Understand the purpose of each of those parts and recognize which parts of the pathway require energy, which parts produce energy, either directly or through reducing equivalent. So it's like NADH or FADH2. For glycolysis, know this in more detail. This is a really typical, people want you to know glycolysis. Identify reversible and irreversible steps. Identify which steps require and which steps produce ATP or reducing equivalents. Identify which steps require which cofactors. Identify which steps differ between glycolysis and gluconeogenesis, and explain the various ways glycolysis and gluconeogenesis are regulated. So glycolysis, breakdown of glucose, and gluconeogenesis, the making of glucose. And so these pathways, it's not just exactly the same in each direction, because you have those irreversible steps that kind of have to be sidestepped. Citric acid cycle. So what I find helpful is if you like color a carbon in one of the starting materials and track it throughout the cycles as it moves through, and you'll see that it's not the carbon that goes into the cycle isn't the carbon that's going to leave that same cycle. So track it through until it goes through, and count how many carbons are present at each step. Some other pathways to learn. Hopefully I'll have a more detailed outline on these later. The pentose phosphate pathway, PPP. This is kind of like an alternative path for glucose used to help make nucleosides and NADPH. Photosynthesis, so making energy in plants. Amino acid metabolism, so we have transamination, basically the passage of amino groups. The urea cycle, which is used to safely dispose of nitrogenous waste. Know like glucogenic versus ketogenic, as well as be familiar with lipid metabolism. And so again, you're going to have to focus on what your teacher thinks is the most important if you're taking this in some course. So make sure that you know those concepts. But these are some of the core concepts that I think are most important to focus on if you're learning biochemistry, for the sake of learning biochemistry at least.